Hey Bio30s, and welcome to the second half of our discussion on meiosis. In the last video, I walked you through the two rounds of division in meiosis, PMAT1 and PMAT2, which basically results in a single diploid cell becoming four distinctively different haploid cells. And this is used in humans for the production of gametes. If you're a man, you're producing sperm through this process. And if you're a female, you're producing your egg through this process. But I did mention yesterday, and I want to get to that today, about how there are two major functions of meiosis. And one of them is reduction division, which reduces the amount of DNA, making the diploid cell haploid, which I pretty much discussed yesterday. But what I want to discuss today is how meiosis creates genetic diversity. How does it create genetic recombination, resulting in daughter cells that are unique from each other and do not look genetically the same as the original parent cell. So there are two ways that this is done in meiosis. The first method is known as independent assortment. And independent assortment, you can look at it basically like flipping a coin. When independent assortment kicks in is pretty much at metaphase 1. So what I've got here is I've got drawn out our same cell that we were looking at in our last video. It's a 2n=4 cell. And I drew this configuration in the video, the last video. You would have seen the greens, I believe, on the one side and the blues on the other. And what the coloration refers to is, in this case, I'm going to say the green chromatids. So these would be two duplicated chromosomes here, which would be a total of now 4 chromatids waiting to separate. These would be the chromosomes that cell had inherited from the mother. The blue ones, on the other hand, are the ones that were inherited from the father. So that's the distinguishing coloration that you should be paying attention to today. Is the green chromatids are referring to genetic material coming from the mother, and the blue ones are representing genetic material that are coming from the father. So when we get to metaphase 1, I mentioned that the homologous pairs of chromosomes, which are currently duplicated into 4 chromatids, which are referred to as a tetrad. So here's my tetrad right there. That tetrad, basically, it's a random determination whether or not the maternal chromatids are on one side or the other side of the cell. So in the case of a 2n=4 cell, there are 2 possible combinations for the arrangement of the genetic material at metaphase 1. In the one case, you've got all the maternal chromatids on the one side going to one direction of the cell, and the male or the father's genetic material, the original from the father, is going the other direction. In the other case, you're going to get a split. You're going to get one parental bit of, father DNA going in this direction with one of the maternal pieces of genetic material going that way. So you can see here there are 2 possible combinations, and when they split up and then we hit to our second round of division, you've essentially got 4 unique combinations of cells that are now going to see their chromatids actually separating in the second round of meiotic division. So in this case, you've got now you've got 2 sets of sister chromatids that are both from the mother. You've got one where you've got both from the father. You've got 2 cases where one pair is from the mother and one pair is from the father, and the opposite. Now in this case, it's the first pair that's from the father. The second pair is from the mother. In this case, it's the first pair from the mother, second pair from the father. But regardless, when all of these meiotic divisions play out in the end, what you're going to wind up with in the gametes, you're going to wind up with 4 possible combinations in these gametes. And in this case, you're going to have a gamete that has 2 chromosomes from the mother. You could have a gamete with 2 chromosomes from the father. You could have a gamete that has the first chromosome from the mother and the second chromosome from the father. And the other combination where the first chromosome is from the father and the second chromosome is from the mother's genetic material from that organism. So what you're dealing with here are 4 possible combinations in that 2n equals 4 cell. And remember, these cells now are all haploid, so they are all going to have the designation of n equals 2. And the slide here that you can see right below me here is going to show you that diagram as well. But there is actually a very easy way to determine the possible genetic combinations depending on the number of chromosome pairs. So in this case, we determined that a 2n equals 4 cell has 4 possible combinations. Well, the reason for that is you can do some math on this, and it's simply going to be 2 to the second power. Because there are 2 pairs of chromosomes, we're just going to take 2 and we're going to square it. And in this case, my answer would be 4. Well, what if I had 3 pairs of chromosomes? What if I had a cell that was 2n equals 6? Well, in this case, my genetic combinations are going to be 2 to the third power, which is going to give me an answer of 8, right? If I had 4 pairs, it's going to be 2 to the fourth, that's going to give me 16, and so on. To put that in perspective, in humans, how many pairs of chromosomes are we talking about? Well, we're talking about 23 pairs of chromosomes. They give us 46 total chromosomes, but there are 23 pairs. So in a human, the possible combinations of genetic material just by flipping a coin at metaphase 1 is going to be 2 to the 23rd power. Now, if you want to know what that number is, that number, and I've got it in front of me, I don't have it memorized, but the number is actually 8,388,608 possible combinations of genetic material in any given human gamete just by the independent assortment of those tetrads when they line up at metaphase 1. So that's actually quite that's incredible. Like that's an amazing amount of combinations just by essentially flipping a coin as to what side of the cell any given a pair of sister chromatids are going to basically move over toward. So that's the first way we can create genetic diversity and genetic variation or recombination in the daughter cells in meiosis. But there is another method which is probably even more important, and I alluded to this in the last video. In the last video, we talked about how when you look at an image of meiosis, you're going to see cells that, or sorry, not cells. You're going to see chromosomes or genetic material that has 2 different colors. And I indicated to you in the last video that represents the fact that there is genetic recombination of the genetic material going on. And this is called crossing over. So at the same time that independent assortment is sort of happening at metaphase 1, and where the cell is just randomly selecting which side of the plane the genetic material is going to line up on, at that same time, there is actually going to be some direct contact with non-sister chromatids. So let me explain that. So here is my diagram again in metaphase 1. Here's my 2n equals 4 cell again. And you can see I've got all my maternal DNAs on one side, my paternal DNA is on that side. That's the way, in this case, the independent assortment lined up. That's just, again, the flip of a coin. But if I look at that up close, this is what it's going to look like. Here's one of my pairs. So I've got my maternal chromatids over here. My paternal chromatids are on this side in blue. These here, so these X's represent sister chromatids. These little Z's over here that are blue also represent sister chromatids. What's going to happen is one of the Z arms, as I'm describing them here, one of these blue arms is going to interact with one of the green arms. And they are going to actually cross over, hence the name crossing over. And when they do that, and here is my little crossing over event, you can see there's almost like they're taking their, imagine here's this one and this one, okay? So here's my green X. Good thing I've got a green wristband on today. That actually works really well. So here's my green arm over here. Here's my Z blue arm over here. And what they're going to do during the line up at metaphase 1 is they're actually going to cross over one another. And what's going to happen is they're going to exchange sections of DNA where they cross over. So when they're done, this is what you're going to wind up with. You're going to have some of this green chromatid. The maternal chromatid is now going to have sections of the paternal chromatids on it. So what we're looking at now is a combination When those chromatids eventually separate at metaphase 2, or in anaphase 2, pardon me, when they separate in anaphase 2, those chromosomes are not going to look like any of the chromosomes that came from the parent, because you're looking at genetic information that is swapping on a chromosome from the mother's genetic material, from the chromosomal material that came from the mother, and the chromosomal material that came from the father. And this is going to wind up providing incredible amount of genetic recombinational possibilities, because depending on where the arms cross over, do all the arms cross over? You can see a couple of examples here of what happens when multiple arms maybe cross over. You can have all sorts of combinations. So alleles that might be coming from the mother's side of the family could wind up on the majority of a paternal chromosome. That way, any gamete could wind up providing genetic material from both sides of a family. That's why you might say, well, I've got my mother's eyes, but I've got my father's ears, right? Because when the gamete was formed, that eventually formed you, that gamete has genetic information. Even though it's a haploid cell, it isn't just bringing over strictly genetic material from your father or your mother's side of the family. There was recombination of genetic material during meiosis that creates unique chromosomes that actually have genetic information from both sides of the family. And that's what crossing over is all about. And here's another example on the slide. You can take a look at this slide right here. It gives you a couple of examples of where we've got multiple arms that are crossing over, and you can kind of see the genetic recombination that's going on there, and that might maybe make a little bit more sense for you as well. One last thing I want to discuss in relation to the separation of genetic material. Sometimes it can go wrong. There are some cases where genetic material doesn't separate properly at anaphase 1 or during anaphase 2 or during one of the separational processes in meiosis. This process is known as nondisjunction. And nondisjunction, I've given you an example here of where, in theory, what should be happening in this cell over here, this 2n equals 4 cell, is both my green sets of sister chromatids should be moving to this side, and both my blue ones are moving over this way. Now, this diagram doesn't obviously factor in any kind of crossing over event that might have occurred, but let's just assume for this example, no crossing over has occurred. Otherwise, you should see little bits of blue on the green and green on the blue, but I didn't actually draw that in there. But just assume for this point that there was no crossing over that occurred. But what should happen is that all the maternal DNA in this case should be moving to one side to form one of the cells, and the other side should be getting all the paternal DNA. But that's not happening here. What's happened is one of the tetrads didn't separate. So one of the tetrads is actually moving all over to one side of the cell before it divides. So what's going to happen at this point is this cell is only going to wind up with one pair of sister chromatids, which will eventually lead to only one, not two chromosomes being present in the actual gametes. That is called monomacy. So in this case, instead of these gametes eventually being n equals 2, they're going to actually be n equals 1. They're going to be missing a chromosome, so they're going to be considered n equals 1. Instead of having two chromosomes, they only have one. So that means that when that gamete, if it fertilizes another gamete, or they come together with another gamete that has a regular number of n equals 2, the diploid organism is going to wind up being a 2n equals 3. We're going to be short a chromosome. In a human, they would be 2n equals 45, would be an example of a monomacy. And I'll get to a couple of examples here in a second. On the other side, we've got an extra chromosome. So these gametes are eventually going to form with they're not going to be n equals 2. These gametes that are going to form are going to be n equals 3. So if that gamete comes in contact with another gamete and there's a fertilization event that occurs, an egg and a sperm come together, the one has a number of n equals 2, this one has a number of n equals 3, the diploid organism that forms out of that is going to be a 2n equals 5. That's not going to be the normal number. We should have a 2n equals 4 organism that results after fertilization. In a human, that would be 2n equals 47. You'd have one extra chromosome. So, and we're going to be discussing a lot of these scenarios, or a lot of these disorders, especially when it comes to sex chromosomes. You can have all sorts of issues that can occur with sex chromosomes. You can have XXX XXY. That there can be these unusual configurations of sex chromosomes that can lead to certain disorders like Turner syndrome, and we'll discuss that more when we get to reproduction. But for now, I'm going to give you what is considered to be probably the most common and well-known disorder that is related to non-disjunction, and that's Down syndrome. And Down syndrome in humans is basically caused by a trisomy. And by the way, I didn't think I mentioned that earlier. One extra. If I didn't mention that earlier, maybe I did, maybe I didn't. Sorry, I kind of forgot there. Getting a little old. If you have one extra chromosome, that's called a trisomy, as opposed to a monomacy where you're missing a chromosome. But anyways, in humans, chromosome 21, if you have 3 copies of chromosome 21 because of a trisomy that happened during non-disjunction, during anaphase 1 and meiosis of the gamete, you're going to get Down syndrome. And Down syndrome is a very well-known disorder which causes cognitive delays and all sorts of distinctive physical features that are fairly well known if you know anyone who has, or you know anyone in your family or friends who has maybe Down syndrome. The rate of Down syndrome actually increases dramatically as the mother gets older. So it's generally well known that it's the female gamete that's going to undergo the non-disjunction normally. Not saying that a male sperm could not have an extra copy of the 21 chromosome if it doesn't divide properly, but generally speaking, it's the age of the mother that determines the rate of Down syndrome. You can see here on the little graph below, it discusses the rates of Down syndrome and how they actually spike heavily as the mother basically gets toward the years of 40 years and older. The rates of Down syndrome increase dramatically, presumably because the mother, the egg, the ovum that's being produced has one extra chromosome. It has an N number of 24, so it's N equals 24 in the ovum, and the egg or the sperm is coming in as N equals 23, which will then give you that 2N equals 47 number. That would, and it has to be specifically with that 21 chromosome. To finish off today, I do want to discuss the 2 main types of meiosis that are basically going to be in humans. And that's going to be spermatogenesis and oogenesis. 2 O's, oogenesis. It's kind of fun to say, but spermatogenesis is basically going to be the formation of sperm. And both spermatogenesis and oogenesis, and I've drawn them sort of side by side here to kind of show you that there are some similarities. There are some obviously some differences, which I'll get to here. But spermatogenesis is going to wind up giving you from one germ cell. Remember, it's those germ cells, those diploid germ cells, that are going to give us our gametes in the end. Basically, that 2N germ cell is going to give rise eventually to 4 mature sperm cells. So this is a standard meiotic process, which we talk about. One diploid cell is going to give rise to 4 haploid, in this case, gametes. In the case of a female, its germ cell, which is called an oogonium, is basically going to produce 4 cells, but only one of them is going to be could be viable as an egg. You're going to produce 1 cell that's called an ootid, which eventually becomes the ovum. And then you're going to have 3 leftover cells, which are called polar bodies. They're basically leftover remnants that basically are all haploid, but they're basically useless. They can't be used to form a gamete. They just sort of are wasted cells. So in the case of female, we're only going to get 1 viable haploid cell at the end. I mean, even though it produces 4 haploid cells, only 1 of them will be usable as a gamete. Whereas in males, the original germ cell will lead to 4 cells, all of which can be viable as a gamete. So let's quickly discuss spermatogenesis and an oogenesis. So spermatogenesis is starting off with a diploid cell called a spermatogonium, and it's going to undergo mitosis. So the first round of division is going to be a mitotic division. So we're going to get a 2N cell is going to become another 2N cell. And anytime you see that in biology, it's always kind of a good thing. If I have 2N to 2N, you pretty much know we're going to have mitosis going on. If I had N to N, that's also going to be mitosis, right? Because again, the parent has the same genetic configuration as the daughter cells. But if I see a cell that goes from 2N to N, that's going to indicate now that it's meiotic division. And remember, we also talked about cells that were polyploidy. We talked about that in the first video of this unit. So I could even have this configuration. I could have 4N becomes 2N. If I see a division of 2 in the genetic material, that usually is going to, well, not usually, it's always going to indicate that there's a meiotic division going on. The last possibility you could see would be the opposite of that, where I have N equals 2N, and N equals 2N would refer to fertilization. So again, the most common ones you're going to see are going to be these ones, right? 2N to 2N equals is going to be mitosis. 2N to N is going to be meiosis, and then N to 2N is going to be a fertilization. But be aware, if the N's equal each other, that's going to be mitosis. If you see a division of 2 in the ploidity of the cell, that's going to be meiotic division. And if you double the ploidity of the cell, then you're looking at fertilization. So those are something that you can kind of keep in mind, and that will play out very heavily when we discuss our next topic, which will be in our next video, which will be alternation of generations, which we'll get to in our next video. But anyways, getting back to here, we've got 2N to 2N, so we know that's going to be mitosis. One cell is going to basically continue dividing. The other cell that's produced basically goes back to replenish the spermatogonium that are in the testes. That way there's always cells in the testes that can continue to divide. If we just kind of had these divide and all became sperm, eventually you'd run out of cells in the testes for this to happen, and eventually you wouldn't be able to produce sperm anymore if you were a man. So you do have to replenish the cells that are divided. So one cell goes back to replenish the parent cell, the other cell continues on with the rest of the divisions. At this point, we're going into meiosis, so we'll go to meiosis 1 now. The spermatogonium, as I mentioned, becomes a 2N primary spermatocyte. That primary spermatocyte now is going to go under meiotic division to become a secondary spermatocyte. At this point, we have 2 secondary spermatocytes. They are haploid, but they are still in a duplicated configuration because the chromatids, the sister chromatids, have not separated yet. The tetrads have, but the sister chromatids have not. So we have to still separate them. That's going to be our third round of division in this case, meiosis 2, in which we separate all the sister chromatids. We wind up with now 4 spermatids at the end. They are then going to mature into what we call spermatozoa, which is the scientific name for a sperm cell. So that's basically the process of spermatogenesis. And here you go. I didn't actually bring that slide up earlier. Here's a slide. You can take a look at it. Missed that slide. It happens occasionally, but you can look at that if you've got your slides on yet home. All right. Last topic before we end today's conversation is going to be oogenesis. All right. So oogenesis, you'll notice that all of the suffixes for all of these structures, they all are the same as in spermatogenesis. The process is actually very similar. You've got spermatogonium, oogonium. You've got primary spermatocytes. You have primary oocytes. You have secondary spermatocytes. You have secondary oocytes, right? So all of these have very similar names, and then these spermatids are analogous to the ootids at the end. The only thing that's different now are these polar bodies, which I'll get to as well. And there's one other thing I'll mention as well that's a little different. The oogonium it's a germ cell, and it's going to start dividing actually well before puberty. It actually begins to divide when the girl, I guess, is actually still a fetus in its mother. Females are producing their primary oocytes before they're even born. So what happens is the oogonium undergoes mitosis, and then it actually begins meiosis 1. It actually goes into prophase 1, and then it stops. So it actually just starts meiosis, and then it stops at prophase 1, and then those cells will basically be in a state of arrested development until the female hits puberty. Then at that point, then certain primary spermatocytes will then kick in to basically become eggs or ovum or ova, pardon me. And that happens, of course, once a month. We'll discuss ovulation and all that good stuff when we get to reproduction in a couple of units from now. But so this primary oocyte is going to then continue meiosis 1 after puberty. It's going to go into meiosis or into metaphase 1, anaphase 1, telophase 1. And then it's going to become a secondary oocyte. However, it's an uneven division. The one cell becomes a secondary oocyte. The other side becomes this sort of remnant polar body, which is sort of like it's an uneven division. We're trying to give most of the cytoplasm, most of the the extra material around this, the nucleus, we're trying to give as much to it as the egg as possible because the egg is actually quite a large cell, so it's going to unevenly divide. Genetic material is going to be evenly divided, but everything else will be unevenly divided. Cytoplasm, organelles, all that good stuff. Now, this polar body, which is useless, may or may not divide into two more polar bodies in the third round of division, meiosis. But the secondary oocyte will again undergo an uneven meiotic division in meiosis 2, which will lead to a third polar body, but it will result in the big ootid, which is basically going to basically be I guess the progenitor to the egg. The ootid will then basically mature into an ovum, but that will only occur if that gamete comes in contact with a sperm cell. And we'll discuss this in greater detail when we talk about conception in the reproductive unit. But for now, this is sort of the way sperm and eggs are produced. They use all three modes of cellular division. They're using meiosis 1, meiosis 2, and mitosis. All three rounds of division play in when we're producing our gametes. That's our conversation for today. Homework or your related assignment for this video is go on to my website or come to see me in class and grab a copy of the Cell Division Meiosis question booklet. And that's what you can do now to basically go over the concepts that we covered in this video and the meiosis part 1 video that you saw previously. Thanks a lot, guys, and we will see you next time.